my observations. Thank you.
Can I comment on that? So there's very little aspartate or asparagine in gluten proteins. But the interesting thing, if you take a step away from celiac disease, is that with the biologic agents that we use, there will be asparagine and aspartate. When a protein ages and deteriorates, one of the typical reactions is for asparagine, so it's a bit like the glutamine story, but it degrades to aspartate. And so proteins change over time, and their immunogenicity changes as well. And one of the intriguing things that I'm just starting to work on now is if you look at infliximab, which is a monoclonal antibody, over time, or even when it's dissolved in the liquid you need to inject it into a patient, you begin to see deamidation. And there's an HLA association with the anti-drug or anti-infliximab antibody response. And it's the same genes as we see in celiac disease. So I think what we've learned from celiac disease could be, at least the chemistry and the genetics, could be generalizable to a variety of medical situations. And we know that many of the important autoimmune diseases share a very strong relationship to actually DQ2.5 and DQ8. And all that's special about DQ2.5 and DQ8 is their preference for these polar negatively charged amino acids at position 4 and position 6 out of the 9 amino acid sequence. So the excitement, I think, amongst the basic scientists is that celiac disease is teaching us broadly about immunogenicity, and it's proteins that the body has been tolerant to, but then through the inflammatory process they have been modified, but there's no established tolerance, and then they cause disease. So celiac disease is now in the lead as the disease that drug developers will look to when they're developing a targeted immunotherapy. And they do that because the peptides are known, and we have the toolkit now that allows the immune monitoring. So celiac disease in Australia, in America, in Europe, patients are being recruited into trials in large numbers. And we're really, this is the revolution of data from very well run, usually double-blind studies. And we're suddenly seeing some of our beliefs were wrong. Histology has been a great learning experience, but there's a lot more persistent villus atrophy when you have really good quality histology and some of these basic things about the immunology. So we're in a very exciting period in terms of understanding celiac disease, but it's actually telling us about autoimmunity in general.
I have a completely different perspective here. I work in the field of microbiome. All the time we are talking about the high intake of gluten is associated with celiac disease. Plus, we also know that non-HLA DQ2 and DQ8, they are also developing celiac disease. We also know that the gliadin peptide, it has high PQ, that is proline and glutamine residues in it. And we are also talking about the role of transglutaminase in the entire process of developing immunity and developing immune response and everything. But the gut bacteria, they have two roles to play here. One, they can trigger the celiac disease. Another, they can also have a remedial role. How? I'll tell you quickly. First, we were also talking about the cohort studies. We don't have many cohort studies as of now. We currently have only cross-sectional studies. What we know that if there is a dysbiosis, that means an imbalance in gut microbiome before the progression of the disease, then it can actually evoke an immune response. And that improper or abnormal immune response can also have a role to play in this. Secondly, gut microbiome also play a very important role in maintaining the gut permeability. If there is an imbalance, again, it actually causes leaky gut or loses the gut integrity, which actually further enhances the possibility of increasing the progression of the disease. The third and most important is there are gut bacteria which can actually break the PQ bond. So if this particular bond is broken, then there is a possibility that we can actually degrade the gluten completely. Again, when we are talking about breaking this PQ bond, there are two scenarios. One, there are some bacteria which can actually degrade this PQ bond and create the immunogenic peptides. And then there are certain bacteria which can actually degrade this PQ bond and create non-immunogenic peptides. So the research is going on. Actually, we are working on this quite a lot in last few years. And let's see, I mean, we can probably have some target available by using bacterial enzymes. Thank you.
Yeah, may I request, thank you very much, panelists, for a wonderful discussion. And in fact, since morning we have epidemiology, environmental factors,